everyone. I'm not even going to phrase this as a question. I assume everyone here has been sick before in their life. But of course, sickness can be caused by many different things. And today I'm going to talk about, very briefly, bacteria that make you sick. So if you've ever had food poisoning after eating bad hummus, that was a bacteria, most likely salmonella, you know who to blame. And if you get sick with a bacterial infection and you go to see your doctor, most likely they are going to prescribe you with antibiotics, specifically for bacteria. Don't take it if you have viral infection, please. So what are antibiotics? It's just molecules that can kill bacteria. So when you're sick and you take antibiotics, the bacteria dies and you're healthy again. Now, in recent years, there's been an increasing concern in the community, which is antibiotic resistance. So it's basically bacteria that evolved ways of not caring about the antibiotic. In many different ways, they can degrade it, they can do lots of things. Which means that if you're sick and you take antibiotics, you're still sick because the bacteria didn't die. And this is very serious concerns. You already have people now that are dying in hospitals of infections we knew how to cure 10 years ago. And to mention cancer at least once, it's expected that by 2050, there will be more death related to antibiotic-resistant bacteria than cancer-related death. And I guess it will come even earlier if all the amazing people we saw today keep curing cancer so well. So the community is trying to develop alternative treatments for bacterial infections that do not rely on antibiotics. And one of these techniques is called phage therapy. I don't know if anyone here has heard of it before. Phages are viruses that infect bacteria. So like any living organisms, bacteria are infected by viruses like we are, like plants are, like fungi are. And for bacteria, we specifically call these viruses phages. So the idea of phage therapy is that if you are infected by a bad bacteria, a pathogenic bacteria, and you take phages, they can't attack you because they don't infect humans, but they are going to kill the bacteria, and once again, you'll be healthy. So, of course, phage therapy is very promising, but it's not perfected yet, and there are lots of technical concerns before we can use it in a broad manner. And today I'm going to talk about a specific aspect of it. So if we take a step back and we think of humans, we are also infected by viruses and bacteria in many things. And these things can kill us. But they don't always do. In most cases, they don't do, right? You've all been sick and you didn't die. So why is that? Well, it's because of something we've heard about already today, the immune system, the set of processes in your body that is responsible for protecting you against infection. Now, bacteria also have an immune system that will also defend bacteria against viral infection. So if you want to use viruses to kill bacteria, you need to understand how bacteria defend themselves. And you need to understand that interaction so that you can modify it if you need, and also that you can choose the good virus to go with the bacteria that you want to treat. And so in the lab of Rotem Sorak, we study the immune system of bacteria, its different components, the way it works, the way it recognizes infection, the way phages manage to overcome it, all of this. So I'm going to focus on my project, of course, which is on a very specific family of immune proteins. So if you think about a fight between a virus and a bacteria, so here the green rectangle is going to be the bacteria, and this bacteria is going to have an immune system that's made of different components, and I'm interested specifically in a family of proteins that I'm interested in because it's a family of proteins that's actually conserved between the human immune system and bacterial immune system. So for all the people doing human immunity here, I'm talking about the NLR receptors or the node-like receptors. They also exist in bacteria, and in bacteria they are called AVS. And for the purpose of this talk, you just have to know it's an immune protein that has two domains. So what happens when a phage infects a bacteria, the phage is going to attach to the bacteria, and it's injecting its DNA inside the cell. And this DNA is expressed to produce phage proteins. Now, the first thing any immune system needs to do is to recognize there is something wrong. You need to know you're being attacked before you can defend yourself. So AVS proteins do this with a specific mechanism that also exists in humans. They're basically going to recognize a specific phage protein that is a marker that there is a phage around. It's something we call pattern recognition, the recognition of a conserved pattern that you only find in viruses, that you don't find in bacteria, and then the bacteria knows that there is something foreign inside the cell. So what happens is that there is going to be physical direct binding between the AVS protein and a phage signal. And this alert is going to be transmitted to the other part of the protein, which is the sword of the system. It's going to be the part that actually defends, actually will attack the pathogen. I'm not going to focus on this today, so I will just say that for AVS proteins that I've studied, we show that it's going to degrade DNA, and this is going to prevent phage infection. So what I'm focusing on today is how is infection sensed. So it's one thing to know that your AVS protein is going to recognize a phage protein. The question is which one? What is the signal? So to know this, very briefly, that's one thing I did. I infected bacteria with phages. Okay, and then I gathered just the bacteria and I broke them open to get only the inside of the cell, the proteins. In theory, in this mix of proteins, I have the immune protein I'm studying, AVS, and it should be bound to the phage signal it recognizes, right? That's how it works. So I can purify that complex and then just study it by mass spectrometry, which is just a technique that I'm going to use to know the identity of the proteins in my purified fraction. And when I do this, so here on the x-axis, you have a phage protein of interest, and on the y-axis, you have how enriched it was in my purified sample. And here you see that there was about 60-fold enrichment for a specific phage protein that's called KSAP1. You don't care about the name. You just need to know that now I care. I have a name for that protein. I know which one it is. So if you think about the interaction I told you about, now I know which partner in the phage it is, and I can model it. So this is just a model. In purple, you have the AVS protein that you see forms a pocket that's going to tightly hug the phage protein that I identify. And now that we know what it is, we can do other stuff. So here I am showing you defense. So I'm taking bacteria, I'm infecting them with my phage, and I'm measuring how well is the infection proceeding. So fault defense is negatively correlated with infectivity of the virus. The higher the defense, the better my bacteria did. So here you see that my bacteria with an AVS protein, they have 1,000-fold of defense, which is great for them. That is because they recognize the pathogen and they have an immune response, and then the bacteria wins and the phage is dead. But now that we know what is the signal, we can also test a phage that was genetically engineered to not have the signal anymore. And when you do this, you see that it totally abolishes defense, because the immune system becomes blind to the infection, and suddenly the phage is winning and the bacteria is dying. So I hope I convinced you that if you study the way bacteria recognize phage infection, you can find ways to modify phages that will manage to kill your bacteria even though it has an immune system. And you can even go further than this. You can identify specific residues of interest. You can identify the spectrum of bacteria that a phage would manage to infect or not infect. And so it's pretty cool. And to finish, I would like to thank you for your attention, thank the organizers very much for this great day, and thank my lab, Rotem, that is here, and that is here also, my PI, and especially Nitsantal and Abigail Stockar-Havihail, who are two other women in science that were really very much involved in this project. And thank you.